Now, let's look at how we use the same setup to investigate the effect of light intensity on transpiration. The equipment you see here is a bubble potometer. Let's quickly run through its key parts and what they do. We have the transpiring plant, or cut chute, sealed into the apparatus with a rubber bung so no air can leak in. This way, the only reason the air bubble moves is because the plant is taking up water. The reservoir holds water and has a tap that lets us reset the bubble to its starting position. The capillary tube contains the air bubble, and a volume scale lets us measure how far it moves as the plant absorbs water. At the end of the capillary tube is a beaker of water, which stops air from getting in and allows us to introduce or reset the bubble. The lamp provides light, and we can change its distance from the plant to adjust the light intensity. That's our independent variable in this experiment. We also have a fan running at a constant setting so wind speed stays the same, and a thermometer to make sure the temperature doesn't vary. Finally, a timer is used to measure how long each trial runs. The procedure is simple. First, note where the air bubble starts on the scale. Leave the setup running for a set period while the plant transpires, then record the bubble's final position and measure the distance it has moved. Reset the bubble if needed, then repeat the process with different light intensities, by adjusting the lamp's distance, to see how it affects the plant's transpiration rate. For this experiment, we'll need a water basin, a scalpel or sharp scissors, the potometer itself, a little Vaseline or oil, a paper towel, a timer, a lamp, a ruler, a thermometer, an electric fan set, and an anemometer. In the diagram, you can see the key parts of the bubble potometer: the fan set to keep airflow constant, the transpiring plant with its cut chute, the reservoir, the capillary tube with an air bubble inside, the volume scale for measurement, the beaker of water, the thermometer to monitor temperature, the timer to keep track of the experiment's duration, the anemometer, the rubber bung sealing the plant into the apparatus, and of course, the lamp to control light intensity. Here's how we carry out the experiment to test the effect of light intensity on transpiration using a bubble potometer. First, cut the plant chute underwater so no air enters the xylem. Then assemble the potometer underwater, sealing all the joints with oil or Vaseline to prevent leaks. Place the setup on the workbench with the capillary tube still submerged in water. Next, dry the plant sleeves so there's no excess water blocking the stomator. Allow a single air bubble to form in the capillary tube, then place the tube back into the water. Position the lamp 10 cm from the plant and let it adjust for about 5 minutes. Record the bubble's starting position, then leave the setup running for 30 minutes before recording the final position. Reset the bubble and repeat the trial twice more at the same light intensity. After that, move the lamp to a new distance, say, 20 cm, to change the light intensity, and repeat the same steps. Continue with additional trials at different light intensities. And remember, when you're testing light intensity, keep all other factors constant, such as temperature and wind speed, so you're only measuring the effect of light. As the plant loses water through transpiration, we can actually see the air bubble in the potometer start to move. That movement directly shows how much water the plant is absorbing to replace the water it's losing. The farther the bubble travels, the faster the rate of water loss, which means a higher rate of transpiration. It's a clear, visual way of showing how much water the plant is taking up to make up for what's evaporating through the stomator. So, by watching and measuring the bubble's movement, we can determine the plant's transpiration rate under different environmental conditions. Our results show that the movement of the air bubble in the potometer changes with light intensity. In general, higher light levels lead to a higher rate of transpiration. As light intensity increases, the plant's stomator opens wider to allow more gas exchange for photosynthesis. This also means more water is lost through transpiration. To replace that lost water, the plant draws up more from the potometer, causing the air bubble to move farther along the tube. At lower light intensities, the stomator opens less, slowing down transpiration and resulting in less bubble movement. So, the distance the bubble travels clearly reflects how light intensity affects water loss. During our 30-minute trials, we saw that the higher the light intensity, the further the bubble moved, because more stomata were open and more water evaporated, driving up the transpiration rate. This experiment makes it clear that light intensity plays a major role in controlling how fast a plant transpires. When light intensity goes up, the stomator opens wider to allow more gas exchange for photosynthesis. While this helps the plant take in more carbon dioxide, it also means more water vapor escapes through the stomator. To replace that lost moisture, the plant absorbs more water, which we see as a greater movement of the air bubble in the potometer. When light intensity drops, the stomator partially closes to conserve water, and the rate of transpiration decreases. Overall, these results confirm that light intensity is a key environmental factor affecting transpiration. Higher light levels lead to greater water loss as part of the plant's normal physiological processes.